Alright, we're going to use a Punnett Square to illustrate how a monohybrid cross should result in an experiment. We've used capital B to show brown because it is the dominant trait, and a small b for blue because it's the recessive trait. So we're trying to find out eye color. Alright, so if we cross two heterozygous genes, you'll set your Punnett Square up as follows. By setting it up this way, you can see that each allele is independently assorting. Alright, from these results we can see that we have 3 that are dominant for brown eyes and 1 recessive blue eyes, giving us a phenotypic ratio of 3 to 1. There is 1 homozygous for brown eyes, 2 that are heterozygous for brown eyes, and 1 homozygous for blue eyes, giving us a genotypic ratio of 1 to 2 to 1. So these are the expected results for a monohybrid cross between 2 heterozygous genes. Right, now we're going to illustrate a cross between heterozygous and homozygous. Alright. So let's say we're crossing a heterozygous brown eyes with a homozygous blue eyes. Again, the alleles are going to sort independently from each other. So this time, the results show 2 brown eyes and 2 blue eyes. The phenotypic ratio 1 to 1. We also have 2 heterozygous for brown eyes and 2 homozygous for blue eyes, giving a genotypic ratio of 1 to 1. Alright, here's an example of a dihybrid cross between 2 heterozygous traits. We use our fly example for this one, so vestigial wings is represented by the V and ebony is represented by E. She's already used the foil mechanism to place the alleles on her Punnett Square. Again, they're still going to assort independently from each other. Okay. Now you're just going to bring all of your traits down and across, just like the monohybrid cross. It can either be completely normal, they can be normal and ebony, can be vestigial and normal, or they can be vestigial and ebony. So we're going to go ahead and go through and pick out our phenotypes. Okay, so all the ones that are normal are anything that has capital V and capital E. So we have 9 normal. That's going to be the start of your phenotypic ratio. Okay, next you can go through and pick out those that are normal and ebony. There'll be a capital V and 2 lowercase e's. So we have 3 of those. Next, we're looking for vestigial and normal. We'll have 2 lowercase v's and capital E. Again, we have 3. And the last is your vestigial and ebony, giving us a phenotypic ratio of 9 to 3 to 3 to 1.